Okay, we have, I'd like to actually introduce Ryan Kennington, who's I guess, Salt Lake native now, living in Salt Lake. And Ryan is an osteo warrior. He worked in the Schiffman lab at one point, and he actually now works as a researcher at the Huntsman Institute, and we're happy to have him here to introduce Dr. Schiffman.
Good morning. My name is Ryan Kennington. I'm an osteo warrior, and I am coming up on my 10th year of being free from this awful disease. Factor 2018 was my first Factor conference, and I just want to take a brief moment to appreciate just how incredible it has been to see how much this community has grown since. So thank you. Now, it was at this Factor conference in 2018 that I've had the privilege of getting to know the moderator for this first panel, not only as a scientist who oozes with passion for his work, but as someone I've been lucky to work under and learn from. So it is my absolute pleasure to introduce this session's moderator, Joshua Schiffman. Dr. Schiffman is the CEO and co-founder of Peel Therapeutics, a biotech company that delivers evolution-inspired medicine to patients. Dr. Schiffman is also a professor of pediatrics and investigator at Huntsman Cancer Institute at the University of Utah. He works as a pediatric hematologist-oncologist at Intermountain Primary Children's Hospital in Salt Lake City, Utah. Dr. Schiffman graduated from Brown University School of Medicine in 2000, followed by clinical training in pediatrics and pediatric hematology-oncology at Stanford University from 2000 to 2008. His academic research and now biotech efforts focus on engineering evolution for patients with cancer and studying animals that are naturally protected from disease, like elephants. Dr. Schiffman focuses on Osteosarcoma and other pediatric diseases with a commitment to impact diagnosis, treatment, and prevention. Thank you.
Thanks, Ryan. Working in our lab is one of the absolute best things to come out of any of these Factor meetings. So it's a real privilege and an honor to be introduced by such a budding scientist and future doctor. Okay, welcome back. The room got a little bit thinner, but that's okay. I appreciate all of the comments. I actually did leave my mascara at home, because I've been here before, so I knew these things happen. I want to just make a few comments before we begin and introduce this session, the biological drivers of osteosarcoma, which will be one of the best, if not at least the first session of osteosarcoma, the meeting this week, or today. So where's Christina? She around, I can't see where she disappeared to, but, oh, there she is. So that was a phenomenal introduction. Right? And I think what we're going to hear today about the biological drivers is about stealing the fire from Mount Olympus. It is about learning and being on the shoulders of giants. And what people in this room, except those from Utah, may not know, there is a mountain range just yonder. And do you know what that mountain range is called? That mountain is called? Mount Olympus. Yes. So after we're done here, we can go up there and steal the fire from up there. Okay, so that was a very apropos introduction. Okay, so welcome everybody. I am going to just briefly take a moment and go through our panelists. We'll introduce them now so that we can move quickly through, and I think the most exciting part is going to be the discussion and the back and forth that happens afterwards, after the session. So everyone's going to speak for about 10 minutes, and then we have time for questions. We're going to start with James Morrow, who's a pediatric oncologist and physician scientist at Dana-Farber, Boston Children's. And then we'll move on to Mathieu Epinette, who's a research technician at the JAX lab at MIT, but who will be doing his PhD at Harvard. I'm sensing a trend here. Dr. Ramesh, a postdoc researcher from the James lab at Johns Hopkins. And that will be followed by a talk by Kelly Gutpell, Dr. Gutpell, a pediatric hematology oncology fellow at Nationwide Children's Hospital, who actually was in Ryan Roberts' lab, so we're going to get a lot of exposure there. And then finally, Dr. Jones. Now, I thought for a while, what can I say about Dr. Jones, except I've known him for many decades now, and I'm looking forward to his talk this afternoon. So I think even though we've got 4 minutes on the clock, I'd rather go ahead and get started so that we have a lot more time for back and forth and discussion. We do have the Slido mechanism. I've uploaded, downloaded, put it left, right, and center on my phone and my laptop, so we'll see what happens. But I encourage everyone to ask questions, and again, I keep coming back to what Christina said. It's really all about the back and forth and learning and understanding and getting ideas from one speaker to another and seeing how we can all work to make a difference and come up with a cure for osteosarcoma. So with that, you are up, James.
Good morning. Really excited to be here, and it feels somewhat appropriate to be kicking off the scientific talks because the project that I'm going to be mostly talking about is really digging into the origins of osteosarcoma. So really, we're interested in understanding how does osteosarcoma start, how does it evolve, and how is that important clinically. I'll try to keep it under 10 minutes. I might skip over some stuff so I don't get flag shamed, but if things come up, we can talk about it more at the end. The framework that we've been using is really one of normal bone development. So I want to spend a little bit of time at the top just to outline the rationale for that. As folks in this room, I think, are well aware, osteosarcoma largely is a disease of adolescence and young adulthood, which is illustrated by the incidence data here on the left of the slide, where you can see that most osteosarcoma tumors really develop in this sort of peak period of linear bone growth. And the bones that these tumors develop in, by and large, are long bones. And more specifically, they typically occur at the growth plates of these long bones. And so on the right of the slide, you can see what that growth plate looks like. The way that this works is you start with a collagen scaffold, and then these stem cells called mesenchymal stem cells come in. They proliferate, they differentiate, and they form mature bone-forming osteoblasts. So the field has sort of known this for decades, but despite that, we really, I think, still don't have a firm handle on what the cell of origin of osteosarcoma is, and I'd argue that really the reason for that is that we don't fully understand bone development. So if you were to look in a textbook, you'd see a figure sort of like the one that I've pulled from this review here, where on the left we start with a mesenchymal stem cell, and on the right, as these cells differentiate, we end up with a mature bone-forming osteoblast. There are sort of a number of individual marker genes that have been defined within this lineage, some of which are listed here, SOX9, RUNX2, Osterix. But at the level of transcriptome, so sort of all of the genes that a cell expresses, I'd argue that we really don't have firm definitions for what these intermediate states are. And I think that that's hamstrung our ability to sort of understand what's going on in tumors. The other thing that makes this challenging is that osteosarcoma, as this group knows well, unlike many other solid tumors, really doesn't have a canonical oncogenic driver. So you can't just sort of differentiate cells, turn on an oncogene, and ask, does that form a cancer? The genomics here are much more complicated than that. Matthew's going to talk a lot about that in the next session, so I'll be brief here. But what we do know is that p53 is commonly lost. And then as a result of that, and probably some other changes, you get this complex genomic rearrangement, whole genome doubling. Of the oncogenes that we know about in osteosarcoma, and you can see here is sort of the list of them. MYC is probably the one that we are most interested in as a community, I would argue. And that's really because of a lot of work that's come out in the past several years showing that tumors that are MYC amplified tend to be more aggressive, they metastasize more, and unfortunately prognosis tends to not be as good. However, we don't understand how either p53 or MYC alterations, or really many of the genes listed up here, impact bone development. And I think that that's an important missing piece of this biology. So the goals of the project, I'll not go through all of this today, but just to give an outline and sort of maybe food for thought for discussion after the talks, really are to define the cell states, an expression program of normal bone development, to understand how osteosarcoma's mutations impact those cell states. One of the things that we're really interested in trying to dig into is whether there are specific cell states or developmental windows that are uniquely susceptible to malignant transformation. And then all of this we're hoping will inform the way that we can then interrogate and understand osteosarcoma biology, in hopes that this is giving us new ways to think about this disease and potentially new avenues for treatment. So to start, I've imported and adapted a system that was previously published to allow us to, in the lab, take primary human mesenchymal stem cells, to differentiate those cells over a few weeks and cause them to become these mature bone-forming osteoblastic cells. And you can sort of see what this looks like at the top of this slide. Over time, the morphology of these cells is changing. You can see that they're laying down extracellular matrix. And then on the right top, you can see that that extracellular matrix is mineralized. So these cells are really forming bone. And then if we look at markers of bone development at the bottom, just by qPCR, we see that those markers are going up. In order to characterize this system more fully and dig in more deeply, we performed single-cell RNA sequencing on these cells over a time course of differentiation, and that data is illustrated here. So here on the, going sort of counterclockwise, what we have are these undifferentiated MSCs, and then day 3, 7, and 14 of differentiation. And what you hopefully can see from this image is that the differentiation of these cells in this system is pretty homogeneous. When we look at cell cycling, we sort of see what we would expect. So these stem cells largely are cycling. At early time points of differentiation, they continue to cycle in this sort of what we were calling a progenitor state. And then at these later time points, as these cells are becoming more terminally differentiated, they largely become postmitotic. And this is all sort of jives with what we would expect biologically in this system. To benchmark the system a little bit more unbiasedly, we've done a couple of different things that I'll just briefly talk about. The first is a lineage agnostic marker called cytotrace, and we see that, as you would expect, these cells look more differentiated, just unbiasedly at the level of whole transcriptome over time in our system. And then using a marker gene expression score, we see that this sort of osteoblast score, as we term it, goes up over time in culture. So this was encouraging, but we really wanted to dig in and do exactly what I mentioned at the top, which was to ask, can we define transcriptional programs that really are underwriting and indicative of these discrete cell states? And so the way that we've done this is through a technique called non-negative matrix factorization. And the idea here is that we look at modules of genes or sets of genes that are coordinately going up or down in gene expression, and then we can look at how the expression of those programs is changing over time. And so this is what that analysis resulted in. So here on the left is our reference UMAP of MSCs to differentiated osteoblasts. And we can see the usage of these programs that we've named here. We define a program that really is specific to our stem cells, a progenitor program, a program that is being used at later time points as these cells are differentiating, and then these more terminal osteoblastic programs. We then wanted to ask, what does this look like, and is this helpful in understanding tumor biology? And so we've done this through a couple of collaborations, the first with Alejandro Sui-Cordero's lab, and some of that data is shown here. So their lab really nicely has defined sort of two flavors of osteosarcoma that are illustrated here. In the middle in green are these EC1 cells. So these are osteosarcoma cells that are aggressive, they metastasize, prognosis is worse. And then on the shoulders, these orange cells are these EC2 cells that are less aggressive, metastasize less. When we look at the usage of the programs that we've defined in the normal system here, we see that these aggressive green cells in the middle are robustly expressing this progenitor program, whereas the less aggressive EC2 cells are expressing a more differentiated committed program, which we thought was interesting. And then we're looking in another way in a cohort that comes from Dana-Farber. So what we're doing here in collaboration with Riaz Jalani is looking at single-cell RNA sequencing data from primary tumors and from metastatic cells and projecting that back into our normal system. And what we see, as you hopefully can see in the bottom right, is that the primary tumor cells largely are projecting onto differentiated bone-forming cells, whereas the metastatic cells are projecting onto stem and progenitor cell populations. And so we're already sort of excited about what this is telling us about biology, but we wanted to, as I mentioned, start to perturb this system and ask, how do osteosarcoma-relevant genetic perturbations impact development? And so the first thing that we did was knocked out p53, which you can see worked relatively well here, and then we take these p53 knockout MSCs through differentiation and compare them to their wild-type counterparts. And what was quite clear was that as these cells in blue, which are p53 null, are exposed to the same differentiation cues as their wild-type counterparts, their transcriptional responses are remarkably different. And we again used NMF to pull out some p53 knockout-specific programs, and I'll just briefly sort of mention, we've been digging into this, but we think some of these programs might be important in maintaining stemness, in cell migration, and then I think potentially most interesting, I think my laser's dying, are these genes that are typically expressed by other terminally differentiated mesenchymal tissues. And so this has sort of led us to think that p53 maybe is a gatekeeper for lineage fidelity in bone development. And we're excited about what that might mean. And then I'm running out of time, so I'll skip over this, but if anybody wants to talk more about MYC, we've also overexpressed MYC, and the punchline is that we think MYC overexpression really is maintaining stem-like programs in these cells. So I'll stop there. I don't think I've been flag shamed yet. And just say thank you to everybody in this room for listening, to the organizers for the invitation, to Edward's family for helping to pay for the trip, to our collaborators, our folks in our lab, and funding sources.
And hold your questions or put them on Slido, and we'll have great big discussion at the end. Okay, Mathieu, you're up.
Cool. Can everyone hear me? Well, thank you everyone for being here today. My name is Mathieu Epinette. I'm a research technician in the JAX lab, and I'm really excited to be here today to share some of the work that we've been doing in characterizing a genetically engineered mouse model of osteosarcoma. And so James nicely reminded us of the prevalence of p53 mutations in this disease, and I think relevant to this talk is the fact that RB1 mutations are also observed in 40% of patients. Another characteristic of this disease are these highly rearranged genomes here, and so in these circus plots, I'm showing you an example of a human osteosarcoma sample. The outer circle represents the relative copy number changes, while the inner lines here are representative of structural variants. And so you can notice by the amount of structural variants here that have probably caused a lot of these copy number changes, that these are highly complex, genomically complex tumors. But the question is, how do we get to this point? How do we get to these complex genomes? And so I think it's been nicely established now that chromothripsis is an excellent source of genomic instability in these complex genomic rearrangements. And so to briefly go over this mechanism, chromothripsis is an event of catastrophic chromosome breakage. And then through DNA repair, we get formation of derivative chromosomes. We can get amplified circular chromosomes. And in most cases, we get fragments that are actually lost to the cell. And we can actually visualize this process using these recon plots here. And here I'm showing you a simplified example of chromothripsis in one of these recon plots. And in these plots, the horizontal lines here represents the tumor DNA copy number, and the vertical lines represent the structural variant breakpoints. And so you can imagine that through these interleaved structural variants and through possibly duplication or inversion events, we've had formation of derivative chromosomes. And in most cases, again, this leads to fragments being lost to the cell, and this is seen by this oscillating copy number here. And this is actually quite a common process in osteosarcoma. There's been a growing appreciation for the prevalence of chromothripsis in cancer in general, And osteosarcoma specifically stands out with around 75% of cases showing canonical signs of chromothripsis. And so our lack of understanding of the impact that these complex genomic rearrangements have on tumor biology has really hindered us in possibly molecularly stratifying patients and establishing targeted therapies. And so in order to get around those issues, in the JAX lab, we turned to a genetically engineered mouse model of the disease. And so the model was first described in 2008 by the Lees and Orkins lab. It bears the conditional P53 and RB1 alleles, and there's a CRE driven by an SP7 or osteoblast-specific promoter. And so deletion of P53 and RB1 in these osteoblasts leads to the development of tumors that histologically resemble a human disease and can seed the lung and form calcified metastases. And so unfortunately, the only piece of genomic characterization that's been done in this model was spectral karyotyping in 2008 when the model was first described, which showed these complex or abnormal chromosomes here, as pointed out by these arrows. And this, to us, was an early indication that this model may be genomically complex and bear some of these chromothriptic events. And so in order to further characterize the model, we performed high-depth whole genome sequencing on some of these mouse tumors. And so we sequenced a total of 23 tumor samples, some of which were primaries and some metastases. The first thing we noticed was that the tumor mutational burden was similar to that of human and, quite importantly, low. And in terms of structural variance, this was high relative to other cancers, and the range that we see in human patients was also nicely recapitulated. And so seeing this range in structural variance, we want to take a closer look at these and see, well, how closely do they resemble what we see in the human disease? And so to do so, we can use these recon plots that I introduced earlier to compare both mouse tumors to human tumors. And so you'll notice again these oscillating copy numbers here and interleaved structural variance in both samples. And just to add a layer of complexity, we also have structural variants that span across chromosomes and involve different gene locuses. And some of you may have noticed that in both of these samples, these chromothriptic events have resulted in the amplification of oncogenes here, MYC in the mouse and cyclin E1 in the human. And this was exciting to us as it showed that, similar to the human disease, these complex genomic rearrangements can actually amplify oncogenes in the mouse. And so here I'm showing you yet another example of these recon plots, and you'll notice again these oscillating copy numbers and interleaved intrachromosomal structural variants. And this time this has led to an amplification of MYC with a predicted 15 to 16 copies. And so we ran Amplicon Architect on these samples, which predicted the formation of extrachromosomal DNA right around the MYC locus. And we were a bit disappointed to see that this wasn't exactly on the gene, but this was something we were able to look at in vitro. And so before isolating DNA to sequence this tumor, we actually re-derived the cell line, which allowed us to perform DNA fish. And so you'll notice here that in this 1535 cell line with a predicted 16 copies of MYC, and you compare this to a control cell line with only 2.1 copies of MYC predicted, you'll notice that there's significantly more of these red puncta signifying MYC amplification. And this was a nice in vitro validation, as it nicely recapitulated what we saw in the whole genome sequencing with an average of 12 to 16 puncta per nuclear region. And when you actually zoom in to these puncta, you'll notice that they're not necessarily associated with the DAPI-positive regions of DNA, implying that these are actually extrachromosomal amplifications. Okay, so far I've shown you very complex cases that we saw in our mouse model with a lot of structural rearrangements, oncogene amplification, but we were also excited to see that this model generates a range of genomic complexity. And here I'm showing you the tail end of this range with tumors that had far fewer structural variants and copy number changes. And I think this range here is actually one of the key strengths of this model, as it allows us to start asking questions about, well, how does genomic complexity affect patient outcome or response to treatment? And given that this can all be done in a treatment-naive setting, we could also start asking questions about tumor evolution and heterogeneity. And so the first question we set out to answer was, given this range, how do these tumors evolve? And so to do so, we performed multi-region sequencing. We had a mouse that developed a primary in its tibia. We surgically resected the primary and allowed the animal to develop lung metastases. At Takedown, we also gathered all the lung mets and sequenced everything at the end, hoping to identify structural variants, copy number changes in these tumors to hopefully reconstruct the phylogeny. And so here I'm showing you a summary of all the structural variants and copy number changes that we identified. And so in this plot, each chromosome is represented by a column. Each sample occupies a row. The individual black lines represent structural variant breakpoints, and the color scheming represents copy number change. And so you'll notice that although a lot of these structural variants and copy number changes are shared across different sample, there's a lot of examples of both structural variants and copy numbers being private to individual samples. And here, highlighted here, are some examples in chromosome 4, 6, 14, and X. And this is pointing towards the idea that these tumors can undergo continuous chromothripsis and undergo continuous structural variations. And this is especially striking when you compare this to another tumor that we sequenced. We performed another round of multi-region sequencing, and here are all the other samples with the primary here and the metastases here. And you'll notice that there's far fewer of these private structural variants and private copy number changes. And so after seeing this, we wanted to take a closer look at these structural variants and see how they evolve in the tumor. And so we gathered all the clonal, private, and subclonal structural variants that we had identified in the previous graph, and we reconstructed this phylogeny here. Oops. And so you can imagine that at some point, all these samples shared a common ancestor with a common genotype here. And at one point, metastases 4 here broke off and started acquiring its own private structural variants, while the now subclonal common ancestor continued to evolve. And at a later point, MET3 split off and acquired its own structural variants, and so on for MET2. And in the meantime, the primary and now the sample that we surgically resected also continued to evolve privately, all leading towards a significant amount of private structural variants being accumulated over this process, and again, providing evidence of ongoing chromothripsis and ongoing evolution in these tumors. And so with that, I want to conclude by reiterating that this model recapitulates key features of the human disease. The tumor mutational burden and the structural variant burden are similar. The mechanisms of genomic instability are similar to that of the human disease. And importantly, this model generates a range of genomic complexity that allows us to start asking interesting biological questions, and putting all of this together, as I'm being waved, putting all this together makes this mouse an interesting tool to study both chromothripsis, tumor evolution, heterogeneity, and how perhaps the immune system may be recognizing these events. And stay tuned for my colleague Jeff Smith's talk, who's sitting right here. He'll be talking about our efforts to characterize an immune response in this model. And so with that, I'd like to conclude by thanking all the members in my lab, Tyler for his continued guidance throughout this project. I want to say a huge thank you to Jeff, who's continuously mentored me over the past 2 years, and as well to Jantha, who did so much work, so much computational work for this project, and has been a fantastic collaborator. Thank you.
Okay. Thank you. Thank you, Mathieu. As Dr. Ramesh comes up, I'll just share with you that Mathieu shared with me that not only is this his first Factor conference, this is his first conference ever. So congratulations, Mathieu. You did a great job. Dr. Ramesh.
Thank you so much, everyone, for being here, and thank you, Christina, for this opportunity. So I would like to begin with a schematic where we see the dorsal root ganglion, which is composed of multiple neurons, and that innervate the bone that is over here. So majority of the bone that is being innervated by TrkA-positive sensory neurons. So what happens is when the nerve growth factor binds to the nerve terminals, that is the trachea, that sends the nociceptive signals to activate pain sensation. What happens in the context, that is in the context of, what happens in the context of a traumatic bone injury is these axons, they do sprout aberrantly. What we see in the thigh one that is in green color, in the immunostaining over here, that they favor the bone injury or the bone healing. In contrast, to the right, what we have is a benign bone tumor, which is also called as a heterotropic ossification, that these axons sprout to favor the disease progression. What we study, or what based on this, we hypothesized that the NGF-expressing sarcoma cells incite the tracheae-expressing nerves, and that apparently sprouts to favor osteosarcoma disease progression. So to recapitulate what we have, we generated an orthotopic model of human osteosarcoma where we inoculated human osteosarcoma cells into the mouse tibia. And what we have here to the left, that's the H&E image, is the tumor cells. And to the right, we have the Thigh-1 expressing nerve fibers that actually invade the tumor. Based on this, we proceeded to show whether the NGF tracheae signaling do dictate the osteosarcoma progression. So we confirmed that the sarcoma cells indeed express NGF, and there is expression of the tracheae positive axons that invade the sarcoma. This was also confirmed in human osteosarcoma cells, where we see the pan-neuronal marker TAP3 that is adjacent to the sarcoma cells. And we confirmed this using multiple cell lines, and the one that we use is 143B, that's on the top left. We also confirmed this using human osteosarcoma samples, where we have high NGF expression in the neoplastic bone compared to the non-neoplastic bone. That's to the bottom right. And we looked at how the pathological innervation is changing compared to the native bone versus osteosarcoma bone. So what you see in panel A is a TAP3 and CGRP that marks the axons in a normal bone. And from panel B to E, we have typified that these axons that sprout in relation to the sarcoma cells are sensory based on the marker that we see is CGRP. And also we do have TH, that is some of the sympathetic nerves that invade the sarcoma cells. Next, we were interested in some pseudospatial analysis where we looked at how farther away are these nerve fibers in relation to the sarcoma cells. And we found that most of the nerve fibers that are marked as Thigh-1 or in NF200, that is in red, were mostly associated with the pseudocapsule, which means that the one that's adjacent, those are the fibrocartilaginous cells that are adjacent to the sarcoma cells. Next, we sought to disrupt this using a transgenic mouse model where we used a chemical genetic approach. So the tracheae mutant mice, they lack the tracheae signaling. Basically, it's hypermorphic, so it lacks the tracheae neuronal signaling. And what happens is, in place of NGF, the small molecule binds to disrupt the neuronal signaling, and that's how we shunt the NGF and tracheae signaling. So what we looked at is, so the tracheae wild type is normal, and then the tracheae mutant is the one that lacks the neuronal signaling. We looked at the growth curve over a period of 4 weeks, and what we found was the tumors not only grew smaller, but also they grew slower compared to their wild type cohorts, as we see in the left. And this was associated with a 52% reduction at the end of the day 28. And the animals did, that is the tracheae mutant animals did survive longer compared to the tracheae wild type animals. Like the overall survival was also improved when you disrupted the tracheae signaling. Next, we tried to look at some phenotypic analysis. So these are some immunostaining that shows that there is reduction in nerve fibers, just to confirm that there is less nerve fibers that invade the sarcoma. And we had a massive reduction in the endomucin blood vessels that were also reduced in the mutant mice compared to the wild type. And the the tumor proliferative cells were also disrupted in the context of tracheneural signaling. We then computationally try to analyze what could be the reason for the decrease in the tumor growth and the improved survival. So we try to perform single-cell analysis from the TrkA wild type and the TrkA mutant mice. The top panel shows that we identified the 2 genotypes of the sarcoma cells, and the panel B shows that we identified the human tumor cells and the mouse niche that are surrounding the mouse tumor niche that are identified by different cell types, as in panel C. We also looked at some gene ontology analysis where we looked at some signaling pathway shifts that were disrupted in the context of tracheneuronal signaling, and we found that the axonal as well as the endothelial growth factor were the most predominant one that we saw when we disrupted the TrkA signaling. We then tried to computationally model, since it has the tumor cells, which is human, and then the mouse cells, which are the other niche cells. So we try to computationally model by looking at how the tumor cells interact with the niche cells. And then we looked, what we did identify was we used CellChat, which is a computational model where we identified several pathways that were deranged upon impairing TrkA signaling. And one of the hit lists that we saw was the neurotrophin, NGF, and the CALCA signaling, which is the CGRP, that were more pronounced when we disrupted the TrkA signaling. And we confirmed this by using immunostaining for CGRP, where the mutant animals had less CGRP expression compared to the wild type. So based on this, we proceeded to see maybe if this is true, then we tried to utilize or repurpose an FDA-approved CGRP receptor antagonist, which is approved for acute migraine. So that's called as Ramesh. So there are different modalities for disrupting the CGRP signaling, and the one that we chose is the third one. That's the small molecule-based inhibition. And what we saw was the Ramesh, or when you basically inhibit the CGRP receptor, we had less tumor growth, and that what we see here was about a 50% reduction in tumor growth over a period of 4 weeks. So this is more mechanistic, that we identify CGRP, and then we inhibit the receptor antagonist, which is on the tumor cells. We also wanted to utilize another modality which is not restricted to one signaling pathway. So we used another FDA-approved lipid nanoparticle, so that's called as Exparel, which is a liposomal formulation, and that's approved currently for post-surgical analgesia. And interestingly, what we found was at a particular dose, it not only inhibits or reduces pain, but also it reduces tumor growth. So this graph over here shows that over a period of 28 days, when we injected, so these are intratumoral injections of L-bup, where we found that the tumor grew smaller and as well as consistent up to 28 days, we found a 60% reduction. And of course, to confirm that they did have less pain, we found that the paw withdrawal frequency, which is Warnfrey, showed less pain compared to the PBS-treated group. So I would like to summarize the take-home messages that NGF-expressing tumor cells incites axon in growth, and that proliferates the tumor cells. And when you disrupt the TrkA signaling, it reduces the tumor growth as well as it impairs the neuronal ingrowth. And when we particularly target the neuropeptide CGRP using the FDA-approved Ramesh, we found that it suppresses tumor growth, and we also used local silencing using L-bup, which is an analgesic that also indeed inhibited the osteosarcoma growth. So this highlights that there are 3 different approaches to target the tumor growth. So this is the summary of what I presented is also available as a preprint over here. And this is my lab. I thank Christina as well as Dr. James and my other, the funding and everybody for this opportunity. And thank you so much.
Thank you. That was absolutely fascinating. I haven't thought about denervation before. That's great. I look forward during the discussion and talking to our orthopedic surgery colleagues about how practical it would be to just cut the nerves. I'm looking at you, Dr. Weiss, so we'll talk about that in a moment. But now, let's talk about anchor cells. Dr. Gutpell, thank you.
Thank you. So I'm Kelly. I'm a clinical fellow at Nationwide, and I'm looking forward to talking about some of the work we're doing in the Roberts lab. So we know that metastasis and osteosarcoma, it's an inefficient process. Many tumor cells enter circulation every day, yet few can actually successfully colonize the lung. So this highlights a critical bottleneck in the process. Previous work in our lab has identified a hypoproliferative, hypersecretory subpopulation of cells that interacts with the lung epithelium and establishes the metastatic niche. This was demonstrated in what we call a red-green experiment that I highlight up here on the top right. So here we have OS17 cells that are sequentially injected with either GFP or mCherry labeled cells. So first, they're injected with GFP labeled cells, and then 2 weeks later, injected with mCherry cells. mCherry cells preferentially localized to regions where GFP cells were seated, and eventually the mCherry cells outnumber that original GFP population. So we became interested in these GFP cells. What is it that allows them to survive the metastatic bottleneck and call those more proliferative cells to them? So we refer to these niche-establishing cells as anchor cells, and functional studies in our lab have shown that specific cytokines, such as IL-6, CXCL8, are key players in this process. In a separate study, down on the bottom left there, our group isolated metastatic tumors at different time points and used single-cell RNA sequencing, which revealed a very clear single phenotype at that early time point. And we hypothesized that this bottleneck may represent a place of vulnerability that we can profit from in terms of potential targets during metastasis. So this previous work prompted us to ask, what is that phenotype that facilitates survival at the bottleneck? We hypothesized that understanding the biology of these anchor cells through single-cell sequencing, we can harness that knowledge to better target them therapeutically. So here I'm just showing the general methodology that we use for isolating our tumor cells to generate our single-cell RNA sequencing libraries. So we use a systemic model of metastasis where we inject OS17 cells via the tail vein, and we pool lung tissue from 10 to 12 mice for further processing. And in that original single-cell sequencing study that I showed you on the previous slide, we used traditional flow cytometry to isolate the stromal cells from the tumor cells at day 14 for our early time point. But when we did this, we had concerns about when we regenerated the data, because when we do it using traditional flow cytometry, the cells are sitting in a cell sorter for at least 6 to 8 hours, and we were concerned that we're introducing potential artifact in that process. So in this study, when we regenerated the data, we leveraged the xenobiology of the model by using a magnetic column sort where we were able to tag murine cells and essentially sort those out so we could just isolate our human cells. And when we did it in this way, we were able to reduce our processing time by about 10-fold. So basically got the cells from lung and got our specimen onto a chip within an hour and a half. So these plots here show our single-cell data after our optimization process, and with each dot representing a cell, cells closer together having similar gene expression profiles. And when we optimized our processing methods, we no longer saw that single distinct phenotype that we originally saw in that previous study. Instead, we saw that a very heterogeneous population of cells exists right at that metastatic bottleneck, and which at a high level, when you compare it to a more advanced or a more established tumor, such as a 28-day tumor, it really closely mirrors what we see there. So while this was unexpected, it was fascinating that we saw this similar heterogeneity at the metastatic bottleneck. And when we looked further at specific markers that functionally had defined our anchor cell population that I show on the right plots there, such as P21 positivity, Ki67 negativity, we didn't see, again, a distinct population of cells. One thing that we did see in the bottom two graphs there is that MCL1 is upregulated at the time of the bottleneck, and this was one finding that was consistent with our previous functional data from our lab. So together, these results suggest that the anchor cell phenotype is not a specific population, but in fact, it's a very heterogeneous group of cells that can adopt multiple phenotypes as they traverse through and survive that metastatic bottleneck. So our single-cell sequencing results suggest that the anchor phenotype, again, it's not significantly different from more established lesions, but we still had data from our previous studies that do show that these cells exert an effect on the surrounding lung stroma. So we wanted to better understand what is it about the stromal changes that characterize this interaction at the early time point between the tumors and the lung stroma. So we have a system that I summarize in the top figure there, where we're able to isolate tumor-interacting stroma. So here we inject tumor cells that express a soluble M-cherry protein that is secreted by the tumor cells in the lung and then taken up by the immediately surrounding stroma. And that allows us to isolate the tumor-interacting stroma based on M-cherry positivity. And so when we do this and we look just topographically at the tumor or the stromal composition at that early time point, which is shown in that middle graph or in the middle plot there, you can see the changes in composition that change between the healthy, non-tumor containing control lung on the left versus what the stroma looks like at that bottleneck. And to us, the most striking difference is in that green population there, the tumor-associated macrophages, which really become enriched at that early time point, as early as 14 days after injection. So based on that compositional shift in the tumor-associated macrophages, we wanted to look at the earliest changes induced within those macrophages, so looking more at the phenotype. And when we performed differential expression and gene set enrichment analyses to look at the to look at the cell signaling pathways, we actually saw very few pathways were affected between the healthy tumor-associated, or the healthy macrophages and the day 14 ones. But in the pathways that were affected, at the very top of the list was collagen metabolism. And when we look at the genes that are affected, we see upregulation of a number of genes that lead to collagen deposition. And so this was consistent with previous work that showed the importance of fibrogenesis in the lung's adaptive response to tumor cells and implicates TAMS as a potential key player there. So though the macrophages had the most striking compositional difference at the bottleneck, we still continued our theme of wanting to look at the fibrogenic reprogramming and next looked at the fibroblast phenotype. And if you recall from the annotated plots that I showed you, there wasn't a huge difference in the composition of fibroblasts, meaning that they weren't specifically enriched. But when we looked at the differential expression and the gene set enrichment, there was actually over 1,100 pathways that were affected between the control and early time point. And once again, at the top of the list, we saw upregulation of a number of pathways related to extracellular matrix deposition and fibrogenesis. To me, one particular interesting finding was upregulation of IL-6, because previous studies in our lab, or from our group, actually show that tumor cells were a primary source of this cytokine, and these findings additionally suggest that fibroblasts might be an important contributor as well. So while we hypothesized that survival through the metastatic bottleneck required a distinct single tumor phenotype, in fact, tumor heterogeneity is preserved even at the bottleneck. And this discovery was facilitated by us optimizing our processing and has really reinforced our understanding that we really need to think about how we generate data sets. And basically, data sets are only as good as the way in which they're obtained. And then, of course, as I showed you at the end there, fibrogenic changes do result from the activity of this tumor heterogeneity, even at the early time points. So now we're going to shift our focus in our future studies to look more at that anchored condition niche and look at outside-in signaling to identify a list of candidates for further study to determine the tumor matrix interactions that occur at the bottleneck to drive the tumor cells to adopt that proliferative state. And with that, I'd like to thank my lab, especially the mentorship I'm getting from Dr. Roberts, and then Dr. Cannon, who's been tasked with teaching a clinical fellow how to code, and then Yogesh as well, who's going to be talking to you later about some of the cell type annotation, and of course, our sources of funding. And I'd like to thank the organizers again for giving me this opportunity to speak today.
Thank you. Another great talk in this. All the other sessions are going to really have to live up to the high standards. Okay, Dr. Jones, my friend, my colleague, my brother from another mother, welcome and teach us something.
Thanks, Josh. Thanks all of you for coming today, and thanks for giving me the opportunity to speak. As you can see, I'm about twice as old as all the other speakers. And I'm actually, in terms of the research that I do in my lab, I am less than half as focused on osteosarcoma. I treat osteosarcoma patients as a surgeon regularly. I did a rotationplasty yesterday to take out an osteosarcoma. I initiated my lab very interested in osteosarcoma, and some of the reasons why I have drifted away from it is because it's tough. It's a very difficult disease to understand biologically, and I want to go into that a little bit. So we're going to begin with some definition of terms. So osteosarcoma genesis, I would say genesis means the beginning of something or how these things are initiating. Osteosarcoma, delightfully, has a very, very simple definition. It's fascinating if you read the literature on this, as I have for the last quarter of a century. People are always trying to come up with some marker. If you publish a paper on mouse genetic models, which I do a lot of, they're always saying, well, what's your marker that you know there's an osteosarcoma? It's like, no, we don't need a marker. Osteosarcoma is defined as ugly cells making osteoid matrix, and that's it. So we know from the very beginning, this definition of osteosarcoma has existed since before probably any of us in the room were born. What it tells us is that cells are pleomorphic, meaning they have many shapes and sizes, and that includes both the nuclear morphology and the cytoplasmic morphology. So these are cells that are ugly in every way. They are different every cell to every cell. So, and then let's define our term epigenetics. So epi just means anything that's around, or specifically not the thing that you're talking about. So the word that it's modifying is gene. Now this becomes the real question. So what are we talking about when we're talking about genetics in osteosarcoma? Are we talking about DNA level changes? Are we talking about chromatin level changes? Are we talking about transcriptome? The definitions of what we consider genetics have shifted a lot over the years. I mean, during, again, my time, even just here in Utah, it has changed dramatically. We used to consider things like microRNAs to be epigenetic phenomena, but just because they weren't protein coding genes. Now we certainly consider those as genes. So how we define our genes really becomes an important question. So we used to define genetics, and in relation specifically to osteosarcoma, as heritable conditions. So doctors Lee and Fromeni defined the Lee-Fromeni syndrome by noticing that there were families that had an excess of sarcoma cases. They also had an excess of other cancers, but they mostly noticed the sarcoma cases. Similarly, just a couple years later, Alfred Knudsen published this really mathematical treatise that was looking at the incidence in terms of age of incidence of retinoblastoma cases. So this is an eye tumor that happens in children, and the incidence of bilateral retinoblastomas versus unilateral retinoblastomas, and made the argument that indeed the bilateral cases happened at an earlier age, and this suggested that these were an inherited predisposition. In other words, that the genetics of this disease, which is another disease that, or condition that certainly includes a high risk for osteosarcoma development later, that the risk of it came in a syndrome, if you will, of those that had bilateral congenital retinoblastoma. So the third syndrome that was linked with osteosarcoma, and this is back in the 70s, is a syndrome called Rothman-Thompson syndrome. We'll talk a little bit more about that in a minute. So the genes for these things started to be identified in the 80s and into the 90s. And this is where we're starting to identify genes as actual coding sequence in the genome. And it wasn't until, the early 90s that David Malkin actually defined the Leaf-Romani gene as being TP53. I mean, that's something that we all accept as the genetic driver of many osteosarcomas, and certainly the osteosarcomas that are associated with Leaf-Romani syndrome, but that wasn't discovered 40 years ago. that was relatively recently. So by the mid-90s, we would say that the TP53 gene and the RB1 gene were loci linked to osteosarcoma. And then it was a little bit later that we finally defined the gene associated with Rothman-Thompson syndrome, which is RETQL4 helicase. We'll talk a little bit more about that in a second as well. So if we look at a different definition of genetics, we would say, are all of the non-familial cases, are those the epigenetic cases, the sporadic cases of osteosarcoma? I don't think we'd define it that way, but maybe we would have back in the 70s, 80s, and even 90s. Another definition of genetics would be if we can make something happen by changing the genes in a mouse. So this is my very old cartoon of knockout mice. So this is not actually knockout mice, but transgenic mice. So where we're moving a gene, usually from a human cell, into a mouse and seeing what tumors it develops. So they noticed that CFOS, proto-oncogene, caused what they called osteosarcomas in the 80s. I would actually say that these were probably driving things more like osteoblastoma, a benign bone-forming tumor. But nonetheless, they thought these were osteosarcomas. Also in the 80s, they noticed that SV40 large T antigen, which we later realized was a protein that's basically binding and inactivating TP53, that this caused osteosarcomas in mice. They then put a mutant TP53 into a mouse and found that these mice developed osteosarcomas. It wasn't until only 20 years ago that we had our first actual models of genetically induced osteosarcomas that weren't just getting lots of other things. And so this is where they're specifically manipulating the TRP53 locus in the mouse in preosteoblasts. And so if we said, okay, now we're starting to profile a lot of human osteosarcomas, and we say, well, TP53 is silenced, and we can silence TP53 in a mouse osteoblast or preosteoblast, and we can form tumors that look like osteosarcomas. Well, does that mean that it's causing it? Then we had these two beautiful papers that were already referenced today by Walkley and Berman in the Stu Orkin's lab and in Jackie Lee's lab that combined TP53 with RB1 disruption in the mouse and found that these formed osteosarcomas very efficiently. And so could we then say, okay, TP53 and RB1 are the genetic drivers of osteosarcoma? Well, what about RegQL4 helicase? It turns out that if you look in human cases of osteosarcoma, other than those that happen in Rothman-Thompson syndrome, they never have loss of RegQL4 helicase. It is almost never observed. And yet, this is a genetic cause of osteosarcoma in this syndrome. What about the other causes of osteosarcoma you can make happen in a model where you hit them with radioisotopes or you hit them with chemical carcinogens that focus on the bone? You will form things that look like osteosarcoma in the mouse every time. What about the osteosarcomas that happen in humans after we've had major nuclear fallout? There was another paper a decade ago that produced DNA damage in a generalized sense across the genome, but in a traceable way. And this was really an elegant study by David Largaspada and Brandon Moriarty. And they discovered that you could either combine it with TP53 loss, or you could just do it by itself in osteoblasts, and you'd form an osteosarcoma either way. So do we really think that these genes drive osteosarcoma, or do they perhaps permit it? If you look at Waddington's epigenetic landscape, this is the idea that a genetically same cell can have very different trajectories of differentiation. And we say that sarcoma is sort of a specific point of differentiation. We induced TP53 and RB1 disruption across a variety of states of differentiation of preosteoblasts and found that any of them were successful in forming osteosarcomas. And in fact, those that developed from the cells that were most differentiated by the time you turned off TP53 and RB1, they were actually the least differentiated in terms of the sarcomas that developed. So we would say that maybe these cells are undergoing a massive genetic change happening at the DNA level, and maybe TP53 and RB1 are permissive of it, probably not causing it. But what is the genetic and what is the epigenetic cause? Is it truly a genetic situation where you have this rampant changes where every cell's DNA in this picture looks different? That tells you that all this heterogeneity we've been talking about in these other talks is present in every cell. Is that a genetic phenomenon? Or what is the epigenetic driver that makes that happen? What are the genetic causes that allow that to happen? I'm just going to pose that question. Thank you.
Okay, wonderful, wonderful job. So we've got mics that are running around with flag bearers if you need them. We've got questions are coming in. It looks like it's working. Before we take the first question, I just want to make a comment that this impact report, if you have a chance to look through, it is incredibly, incredibly impressive. All of the science that MIB is driving and all of you are helping to contribute to. And we've got some questions. Dr. Weiss.
Good morning. Kurt Weiss, Pittsburgh. Josh, Christina, amazing session. This was really, really, really terrific. So, and I'm super, super excited to see all of the awesome work that's going on looking at metastasis and looking at those, specifically those matched sets of the primary and the met from the same person or the same critter so that we might be able to reverse engineer the process of metastasis. So that's what, because that's what's killing our patients. So great job, everybody. And using that as a jumping off point for Dr. Ramesh. So when you disrupt the TrkA signaling, what does that do? Does it do anything to the metastatic potential of that tumor? And sorry, hard left, same investigator, but different question. Should Steve and Kevin and Tom and I be injecting bupivacaine before or after or both when we take the osteosarcoma out? Because that's not hard. Should we be doing that?
Thank you for the question. So I didn't have time to show that, but we did do lung mets. So after impairing the tracheoneuronal signaling, we harvested the lungs from both the wild type and the mutant, and we did see less mets in the when we impair the neuronal signaling. But what's something that we are interested in is to delve more deeper. We just know that we have less mets. That's about it for now. But I think right now I'm inspired by what Kelly has done with the anchor cells to more, to sort out like how the met cells are different by harvesting them probably. But we haven't looked anything more than less mets in the when you impair neuronal signaling.
Hi, John Healy from Sloan Kettering. Congratulations on a great symposium. Kevin, should this be called osteogenic sarcoma, not osteosarcoma?
I like that.
Yes, thank you. And Kelly, would you comment about other things that influence the metastatic niche, like exosomes and the related elements?
Yeah, so I guess right now I was more talking about some of like the cytokines and everything that comes like from the outside in, but something else that we're gonna start to look at is like other factors in like the extracellular matrix, like the like stiffness and like other inputs like that is gonna be a next focus. And also thinking about maybe neuronal input as another input is totally fascinating to me as well. So.
And to follow up on that, Ramesh, what do you see this in other metastatic cancers, for example? Is there neuronal innervation? And just as an observation, BMP is actually bone morphogenetic protein and is more of a neural genesis than a bone genesis. So there's an interface here. Interested in your comments.
Yes, so that is true. So right now we are kind of swaying away from other soft tissue sarcoma as well as breast cancer and prostate, which indeed have, so basically the NGF is what drives the expression or insight or recruiting these axons. So most of the cancer types, the solid tumor types, they do express NGF. So yes, indeed there is relevance of this pathological sprouting that we can observe in other tumor models as well. But yeah, I think that's a good point about BMP, because when we looked at putative candidates that may have contributed to increase in the osteosarcoma progression, BMP is one of the neuronal signal that is a potential candidate too. But right now what we found was CGRP, because that's more abundantly expressed by the tumor cells. And upon inhibition, we found less tumor. But yes, it is true that it is just not one tumor types, but like as Mathieu also pointed out, that the heterogeneity is so wide that it is just not one target that we are looking at. That's why we used LBUP, which it was a serendipity that we found that at particular dose that it inhibits neurite in growth. If that answers your question.
Let's go to a Slido question. It's just fun to say, Slido. Okay, James, how can your model inform diverse populations in osteosarcoma? Is there evidence of multiple differentiation states within patients' tumors? And do you have data to understand different developmental states during puberty compared to earlier development?
And I think that came from Rosie. So hi, Rosie, long time. I'm not sure that I understand the diversity question, so maybe you can clarify that for me. To the second question of whether there's evidence of different states of differentiation within a primary tumor, the short answer to that is very much yes. I didn't really have time to present it, but in the collaboration we're doing with Riaz Jalani, we have sort of a nice cohort of matched primaries and mets, and we've done clonal tracing. So by DNA changes, we're able to identify clones in primary tumors that are also present in the mets. When we stratify those clones by their state of differentiation, we think that's actually predictive of how metastatically successful they are. And then conversely, when we see primary tumors which are enriched for more differentiated states, those patients tend to develop metastases just by reviewing clinical data less frequently, and the opposite's true in tumors that are enriched for less differentiated states. There's not a great reference for understanding what's going on in puberty, so the Human Cell Atlas is working on this, and they just put out a big publication looking at embryonic bone developments. That's sort of the best that we have right now. The way that we've thought about doing this, and maybe for the orthopedic surgeons in the room, if anybody's interested, what we would love to do is to take tissue from a limb length discrepancy surgery where we actually, I think you guys, access the growth plate and remove some of that tissue. I think that would be really an amazing resource for the field to generate atlases there.
That's awesome. I think that answered everything. The only other question that I had that I think you didn't understand was the diversity in terms of the differentiation status. So one person's tumor may have multiple tumor cells that are at different stages of development, and what that diversity looks like in your models compared to patients. That's what the question was.
Yeah, I think I addressed that.
I think you answered it, so thanks. It's really cool.
Great.
Hi, Jeff Smith from Dana-Farber. Dr. Ramesh, this is for you. I was really intrigued by the TRK result in the hypomorphic allele. Have you tried to reproduce that with larotrectinib or any of the clinical TRK inhibitors? Because there would be a fast translation opportunity here.
Yeah, I think that's a great question. So that's one of our pipeline studies that we are considering larotrectinib and entrectinib, which two are the currently FDA-approved TRK inhibitors. And I'm sure, I think that will be fantastic to see that results as well. But as of now, we do not have that results yet, but it's in the pipeline.
This is another question for you, Sowmya. Is there an application to your research for the pain after the resection that patients are dealing with, either in terms of understanding that pain or potentially mitigating that pain?
Would you like to repeat the question? Sorry.
Sure. So I'm just curious if there's any application of your research to better understand pain that cancer warriors have after the resection, and also potentially to mitigate the pain.
So that's it's a very tricky thing because what we did so far is in mouse model. So to directly translate to what a human warrior, osteo warrior would experience is something that we haven't gone much deeper. But because, what we just do is some sort of an assessment like Von Frey and Hargreaves, which is like hyperanalgesia. But so both CGRP inhibitor, receptor inhibitor, as well as the L-BUP, we did see some sort of a reduction in pain. But what stage in which the human patients experience pain, because at some point the nerve fibers are disrupted in that area, so they would no longer sense. So that's a key transition to sort of understand to understand like post treatment pain. But I think that's a good point to consider in case we try to move forward for clinical trials, I would imagine.
Great. We're going to do a Slido for Mathieu. So do the same manipulations driven off of different Cre promoters, the other tissue types, cause similar chromosomal instability, or is there a susceptibility of this osteoblastic lineage to these structural changes? If so, why?
Well, thank you, Ryan, for the question. It's a really interesting question. I don't think I have a very specific answer to it. The only other promoter that I'm aware that is used in this model is the CAL10A promoter, which is a little more downstream in terms of osteoblastic differentiation. I'm not aware of any whole genome sequencing data that has shown similar heterogeneity and chromosomal instability in the model. So I can't really answer that part of the question. And to answer your second question about is there susceptibility to this osteoblastic lineage specifically, again, not having seen what the other model looks like, my only hypothesis as to why it would be different is the fact that this, the SP7 promoter is upstream of the cultinate promoter, and you may have different chromatin remodulation, which allows for cells to undergo these structural changes.
And Ryan, you can check back with Mathieu after his PhD, and I'll have more of an answer for you. All right, great job. Let's do 2 more questions, and then we'll get on with our break. So, please.
Okay, I have a question for James Morrow, and this is going to be more of like a question you get from an undergrad because I'm just a beginner here. But it has to do with differentiated and undifferentiated. And correct me if I'm wrong, but I believe you said that the primary tumor originates from the differentiated cell and the metastases from undifferentiated cells. I just want to know if that was correct, and if that is true, would treatments differ between the two if you're going to target primary tumor or metastases?
Yeah, thanks for the question. It's a really important point. I think also it gets back a little bit to Rosie's question earlier. And I was sort of moving quickly at this point, but when we map tumor single cell expression data onto our in vitro differentiation data, what we see is that primary tumors have an enrichment of a population of cells that express genes that in our model and in, we think in some other benchmarking data sets, are expressed in sort of more differentiated cell types. There are certainly less differentiated cells also within many of those tumors, so it's not really a binary delineation, but there seem to be more differentiated cells in primary tumors and more or less differentiated cells in metastatic tumors. We're really interested in thinking and sort of digging into like the mechanism here, and I think to understand how this could or should impact therapy is going to require a lot more work. But I'm hopeful that if we can kind of convince ourselves, one, that this is real, and then 2, understand kind of what are the mechanisms at play, we could then think about ways to intervene, because in many different data sets now, this phenomenon that we've observed is repeated. So I'm hopeful that someday we'll get there, but I wouldn't necessarily recommend any changes in therapy at the moment.
There. All right, last question.
Yes, I was diagnosed with
telangiectatic osteosarcoma.
So it's super rare. I feel like I have no information. It's so very frustrating. It happened 10 years ago, so I'm grateful to be here. I'm grateful to be alive. Anywhere I can go to gather some information, I feel like nobody knows anything about it. Not nobody, but it's so rare.
It's rare. There are a number of subtypes of osteosarcoma. I think the key, as we heard from Dr. Healy, they all make, so maybe we should call it osteogenic osteosarcoma. Even telangiectatic osteosarcoma would not be called telangiectatic osteosarcoma if they didn't find some part in the tumor that was osteoblasts or malignant osteoblasts making osteoid matrix. And so it is a particular type of osteosarcoma that has a lot of vascular differentiation apparent in the cells, but mostly these are just ugly cells, and some of them are making osteoid. And so what's interesting about telangiectatic osteosarcoma, if you look at it clinically, is that it is actually a little bit better behaved if we're treating it. In the days before we were treating almost all osteosarcomas with MAP chemotherapy, telangiectatic osteosarcoma was one of the worst actors. It was one of the most rapidly progressive and metastatic types of osteosarcoma. But now that we treat it, it actually has a little bit better prognosis than even what we would call conventional osteosarcoma, which is mostly osteoblastic.
Great. Well, big round of applause for all of our speakers. What a great way to kick off the panel discussions with the biological drivers of osteosarcoma. Now we get to have a break until 10:45 when we'll come back and we'll talk about collaborative data ecosystems. So load up on your coffee and we'll see you shortly.